to genetics, so they might see, they might, they know something. Usually from the books, they learn and they
Yes.
know the heritage, whatever
Yes.
disease, autosomal, autosomal dominant,
Dominant, dominant,
recessive,
recessive. Yes.
whatever is more common here and there, but they usually don't apply in the general.
Practice. Yes. Yeah,
So
this is
this
common everywhere, I feel so. Worldwide.
this is always very important
Yes.
for everybody. And you have a busy clinic back with in Aditya Jyot Hospital. You have scheduled twice a week or three times a week for residents and fellows to watch lectures and talk about genetic things.
Normally, we had that practice initially. So, as and when the fellows they keep on changing, so it got disturbed actually. So the continuity is broke.
Ah, very good.
Yeah.
And I saw you deal with more stuff that comes from genetics, like twinkle eyes and glaucoma and syndromes and all things related. And so people probably refer patients to you from their diseases. And what you get most of genetics, we get most of RP, for example, retinitis pigmentosa.
Okay. Yeah. So that we have more actually in our population, especially our clinic receive lot of RP patients, followed by Stargardt's, then retinal degeneration. Basically, I worked for long period in cataract, congenital cataract.
Oh yeah, in cataract as
Yeah,
well.
in cataract. Yeah. So after joining Aditya Jyot, I started working on retinal diseases, especially RP and AMD. Now we are working on diabetic retinopathy as well.
Yeah, but things have changed so much. Those years
Yes,
with so much innovation and
yes, yes.
genetic
Yeah,
medications.
yes,
Expensive, yet so
yes.
expensive.
Yeah.
But
After this invention of next generation sequencing, the things have become a bit easy actually. So in terms of genetic testing and diagnostics. So initially we have to struggle a lot to identify a gene of interest or to identify a variation that causes disease. But now with the advent of next generation sequencing platforms, so it is a bit easy to catch out that particular variants.
yeah, yeah, I know. I remember back few years ago, not very long, so um, I follow. I did my, that was many years ago. I did my retinal fellowship in Canada, and
Oh,
Dr.
okay.
Divini, with Dr.
Uh,
Divini, Dr. Whitechim Lam, Dr. Berger, Dr. Harvey, and
oh,
Dr. Divini. This was not long before he was on Argus 2 implant
okay. Okay.
for RP, and he was quite successful. I think he was the one that did more RPs than anybody in the world,
Yeah.
maybe 19 or more. And
Okay.
because of the financial problems from the laboratories and distributors, and it seems that the Argus 2 stopped for a while. I don't know whether they're gonna uh, get it back, and it looks that they wrapped it up for a while. I don't know whether they're gonna return with the studies and the tests.
Okay.
They were trying to enlarge the electrodes and do better, better
Okay.
stuff. Uh,
Oh,
we actually have in the internet this patient from India that she got very known in the world. and she had a very severe retinitis pigmentosa.
Pigmentosa. Oh.
After the implant, she was wandering around
Mhm.
uh, India. I don't know which city she lives at, but
Okay.
that
Oh.
changed her life forever.
Oh.
That changed her life.
Oh.
That
Okay.
was
That's
interesting.
great. No, that's great.
Yeah. And then, recently these medications that were studied, they came up, and now we have Luxturna that I
Yeah.
don't know any cases here in Brazil, but it's a little bit expensive. And I think
Yes.
I don't
Yes.
know
Very expensive. Yes.
yeah, your idea on that, because
Yeah.
now with the COVID times and the exchange rates for the medications and for everything, actually, things got more and more difficult to follow.
Yes.
So I'm not quite so sure whether uh, this approach will start or
Yes. Sure.
take
Yes.
off better. Understand what I mean?
Yeah.
But
As far as I understand, the Luxturna is not yet launched across the world. It's very limited to certain part of world.
yeah, yeah, I know.
Yeah, and because of the cost as well as the production rate, and it depends upon the patient population again. So how many are having that particular RPE65 gene mutation? Because many RP patients do not have RP65 gene mutation, especially in our case. We have done around 30 cases. None of them had a mutation in RP65 gene. So it means those patients will not be eligible for such trials or such treatment.
Okay,
Yeah.
yeah. And there are more and more studies, and I think we could have an example, as you have from a genetic clinic as well, so that we could also give the example for other residency programs and such as ours, so
Yeah.
that we could direct better our patients on that purpose. And do you usually ask the patients, for example, if you detect somebody that was referred with RP, how do you approach them calling more members of the family and then gathering more people to have the IXM made? And people usually like the idea. They bring the family, they bring everybody, or they find some. You find some resistance on that.
Yeah, yeah. Some patients will be willing to bring in their family as well. Suppose if a proband seems to be a kid, 5 years, 6 years kid, so obviously the parents will accompany them. So only thing, we may not have their grandparents, the proband's grandparents. They might be living somewhere in India or somewhere in the world. So bringing them together will be a bit difficult in the first go. So once we diagnose them of having some diseases, then we take them for a genetic counseling. And we will
Okay.
tell them what are the possibilities of genetic testing and what they can get benefit out of it once they perform or undergo genetic testing. Then some patient or some parent will accept immediately, because if they plan for next kid or next generation, then they will obviously go for it. If some people, some parents may not go for it immediately. So whoever is willing to go for genetic testing, then we can insist them to get their parents also. So in that
Mhm.
case, we can find out the hereditary pattern, because we need to know whether it is a dominant or recessive, or how that is going to segregate in the family. So what are the chances of other members to get affected? So all those possibilities we can share with them. So in that aspect, they will come forward for a genetic testing.
Yeah, yeah, this is
Yeah,
interesting.
yes. So we need to have a genetic counseling, especially when we diagnose them of having some rare or unique phenotypes.
Hmm. And do you get patients from the countries around you, like Pakistan, like other countries? That even from China, you have the not
Dr.
very
No,
far
no, no,
from.
Dr. Natarajan had a lot of patients across world, but there may not be frequent visits of the same patient. The moment when they come into India, then they will just check in and go for the ophthalmic evaluations. If they find something else, then they will go to their place and they will take further follow-ups. They may not come back to Aditya Jyot again. So
Mhm.
following those patients will not be a good idea because we cannot trace them in future. We cannot have samples from their family members in future. So we have not got, and moreover, there is ethical issues also. So we have ethical clearance for our Indian population, not for the outsiders. So we need to get ethical clearance from their country if at all we want to collect a DNA sample from others.
Mhm. Yeah, that's very very interesting,
Yes,
and
yes.
I think that I'm already live on. We are already live on YouTube as well,
Okay.
and this lecture of wars of yours that I
Okay.
thank you very much for accepting the
Well,
invitation.
thank you.
And this is very precious for us to have somebody speak on such a very interesting and great subject that is genetics. And we really appreciate, and this lecture will be recorded and will be there for future research from the residents and
Yes.
fellows. And it's good that we chat before, because then we get to know each other, and
Yes.
also we get to spread the good news for the people that
Yes.
how your subject is, and your subject is of utmost importance. We know that, and
Yes.
I would like you, if you could possibly share your screen and show your presentation as well.
Okay.
I already made you co-host. Hi, Ashwara. Good morning. How are you doing?
Hi, sir. Good morning. How are you?
Yeah, I'm fine. Good to see you. You know, right now some 7 is approaching next
Yeah,
Saturday.
yeah. Gearing
It's,
up for that.
it's getting there.
Right.
Thanks
Hello,
for.
Rajkumar, sir. Good
Hi.
afternoon.
Yeah, good
Oh,
afternoon. How are you?
I'm fine, sir. How are you?
I am good. Hello, Rodrigo. Hola.
Hello. Olá. Good afternoon. How are
Good
you?
Hola,
afternoon.
Rodrigo.
Olá,
hello,
Clara.
Rodrigo. How are you?
Hi, Dr. Nakamura. Fine, and you?
We are here. We are very happy to have Dr. Rajkumar talk about genetics. We know we don't have this specific subject in our teaching program, for example. So it's going to be great. You know, we are very happy to have him uh, speak on that.
Okay. Yeah, thank you all for accepting my talk. I think I have shared the presentation. Are you able to see?
Yes,
Yes,
sir.
it's perfect. Yeah.
Yeah. Okay. Yeah, I have just made some changes in the title what Dr. Aishwarya suggested. So I wanted to keep it very simple and very basic so that even a layman can understand the concepts. So I will be talking about the basic concepts and some application in ophthalmology, and I will take some reference from my own publication. So I may not talk about a lot other publication from other people, so that will be a bit difficult to understand to go with. So let's begin with the contents. So I'll be taking you through these topics like traits and classifications. Basically, I divided this into basic concepts and applications. So under basic concepts, you will be talking about traits and classification of phenotypes, definition of genetics, mode of inheritance of phenotypes, genome and the gene structure, genotype and phenotype correlation, and the genetic variations or mutations. And under applications, how to draw a pedigree from a family history. Then what are the different genotyping techniques are available to do genetic testing? And I have shown one of the example from my paper. Then what are the limitations in genetic diagnosis? So we will be discussing on those things. Okay, so many of us might ask such questions. Why are we alone getting more mosquito bites while some other are not? And why I am so thin? So why is other people are so thin? Despite having and the stuff what is not suggested to eat. Why me alone getting diabetes? Why not others? Why do people respond differently to different drugs? Why I am responding to drug or why I am not responding to drug? So what are the medicines that are suits you? So these are the certain questions. Other than that, there are a lot of other things, but these are the basic things that we can ask. So what could be the reason why we are behaving differently to different things? Why one individual is responding to something, why others are not responding to something? Why we are getting disease, why others are not getting disease? In the same environmental condition, in the same population. So there could be something. So we are gonna study that tribe, what is happening or what is causing that tribe. Okay. So these are the traits called phenotypes. When you look into that previous slide, so here I have marked a few things as phenotypes like mosquito bites. Are these the real phenotype? Mosquito bite is a phenotype because we know there is a mosquito bite just to by the swelling. So that is a kind of phenotype, a non-invasive phenotype that we can see. But is this the real phenotype that we are looking into? What makes that mosquito to bite only a particular person? So these are the things. So there is a hidden phenotype in this. The mosquito bite or the swelling of that area is the visible, observable phenotype, where the actual thing which attracts the mosquito is the real phenotype. So what could be that phenotype? So in general, it is said that a person with O blood group are more prone to have mosquito bite than A and B blood group. So what does it mean? It means the marker lies somewhere in the O blood group. So that attracts the mosquito. And similarly, some patient will be very thin despite having lot of food, despite having all junk food. So their genetic makeup makes them or keeps them very thin. Some may be very fat despite not having anything. So these are the things that is regulated by our internal gene or genetic makeup. Okay. So in genetics, the term phenotype refers to the composite observable physical properties or trait, so which we can easily see or which we cannot easily see through invasive method we can observe. So these are the things called phenotypes in a clinical term. So this includes either morphology, a gross appearance of an individual, or appearance or physical form of a structure, any anatomical changes, developmental process. Some may develop, some may grow tall, some may grow short. So the developmental process, biochemical and physiological properties, behavior, even behavior, and the products of behavior. So all are suggestive of phenotypes. So anything that changes from time to time, so that can be observed. That could be due to the regulation of certain genetic elements in our body. So the traits of phenotypes could be either visible to naked eyes or non-visible to naked eyes. So that can be observed only through certain experimental or evaluation or some diagnostics. So based on this, we can classify the phenotypes. So initially people used to have a kind of phenotypic observation based on the quality, superior quality or inferior quality. So how in the previous era, 17th century or 18th century, normally after the civilization, so people started having a domestication of animals, cattle, equines, all those things. So in order to select the suitable variety, so they keep the superior variety or inferior variety based on their strength, the effect, function, all those things. So they started classifying the phenotype based on the superiority or inferiority quality. Then, when coming to the health sciences, so how we are classifying either a person with normal condition or a person with abnormal condition. The abnormal condition, what we call it as a diseases over here. Okay. So then again we can classify that phenotype into qualitative or quantitative. So what do you mean by qualitative? So anything cannot be measurable is qualitative. So just by the morphology or change in structure, change in the dimension, change in lifetime, change in the strength, force, all those things comprise of qualitative phenotype, whereas that can be measured in an experiment, laboratory condition, like number of blood counts, level of hormone, level of activity of enzymes. So these are the called quantitative phenotypes. Okay, so then the genetic comes into that. So to study the inheritance of such traits from one generation to other generation. So that is the study of gene or study of inheritance. So genetics is nothing but study of inheritance. So inheritance means the way that certain traits or conditions that passed from one generation to another generation. So we call Dr. Gregor Johann Mendel. He is an Austrian monk. So in the 1860, he first noted the inheritance or segregation of certain traits of the pea plants. So he found out that wrinkled versus round pea or yellow versus green pea. So he observed that that particular trait that is passing from one generation to next generation. So that's why he is considered as a father of modern or classical genetics. So he said that there is some trait that inherits as a unit. So that is called unit of inheritance. Still we follow. So although the term is quite ambiguous, but still we consider that unit of inheritance as a gene. So it means the unit of inheritance is referred to as a gene. Okay. So based on that, which I mentioned earlier, qualitative and quantitative phenotypes, we can classify genetics into qualitative genetics and quantitative genetics. So these two have two different, have some differences. Like qualitative genetics, it always deals with the inheritance of traits of kind, see one kind, either form or structure, color. It could be due to single gene variation, whereas the quantity of quantitative genetics could be due to multiples of genes or polygenic. So height and length, numbers of cells, all those things. So then discrete phenotypic classes occur which display discontinuous variation. Means the variation will not be changing every time. So you'll have only one particular phenotype and one particular variation. Whereas in quantitative genetics, you will have spectrum of phenotypes. Say, for example, if you take RP, retinitis pigmentosa, we have 89 different type of retinitis pigmentosa. So that is a variation that is due to the variation with different genes and in the same gene variation at different alleles. So these are all called quantitative genetics. Each qualitative trait is governed by two or many alleles of a single gene, which I explained. Same way in the quantitative, it will be governed by multiples of genes or multiples of alleles. Moreover, the phenotypic expression of a gene is not influenced by environment in qualitative, whereas environment plays a major role in quantitative genetics. That we call it as a epigenetics. Then the qualitative genetics concerns with individual mating and their progeny. So it depends upon a single family or inbreeding or outbreeding, but whereas here it considers the population, polygenes in a population. Then analysis, analysis is made by counts and ratios, whereas in quantitative made by statistical method. All statistical method needs to be applied for quantitative genetics in order to understand the outcome of the genetic study. So, how these traits are inherited? What are the patterns? How it is coming from one generation to another generation? Can we see the same phenotype in different generation or in a subsequent generation? So, based on that appearance of that phenotype, so we can classify the inheritance into 5 different patterns. One is autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive, as well as the mitochondrial inheritance. So in autosomal dominant, each affected person has an affected parent, definitely. And moreover, in every generation, you will have at least one individual affected. Whereas in autosomal recessive, both parents of an affected person are carriers. The parents may not express the disease, whereas the proband will have. Not typically seen in every generation. So it means it skips the generation. You may see in first generation, it may not be there in the second generation, then it will express again in the third generation. So always the autosomal recessive inheritance will skip one generation. Then in X-linked dominant, the females are more frequently affected and they can have affected males and females in the same generation. So it means it is common to both male and females. Whereas in X-linked recessive, males are more frequently affected because the female gives one X chromosome, affected chromosome to the male. So always the male will have that condition. And in mitochondrial, since this pass from mother to the offspring, so almost all individuals in the generation will have that mitochondrial based inheritance. So can affect both males and females, but only passed on by females. So can appear in every generation. So these are the different things that we need to look into the inheritance pattern to decide what type of inheritance that family has. So to draw that, it is better to have a pedigree. So I'll come to that part later. Okay. So before coming to that inheritance, to understand the inheritance, how we can find out the causative gene or causative mutation that is responsible for the disease, we should understand our genome. So our genome, what is our genome? So we have cell. In that cell, we have nucleus. The nucleus has nucleic acids. So the nucleic acids are nothing but the genome. So we have a complete set of instructions that are responsible for making one complete organism. So that is called the genome. So the genomes are divided into chromosomes. So as a human, we have around 22 pairs of autosomes and one pair of sex chromosomes. Either it could be XX or XY. So apart from reproductive cells, and blood cells, RBC cells. Almost all cells are having diploid chromosomes, 2N. So we have 2 pairs of chromosomes. And each strand of these chromosomes are compact in a form of nucleosome that is bound by histone proteins, where histone protein, that is the protein that is responsible for condensation of the chromosome, because the chromosome is a huge molecule. In order to make it to a compact one, so the nucleosome four proteins called histones are playing major role. So to make it very tiny particle. So each chromosome is made up of DNA, deoxyribonucleic acids. The DNA is a double helical strand made of four basic nitrogenous substances called guanine, cytosine, adenine, and thymidine. So each nucleotides, there is a pairing phenomenon or mechanism. Always the A, adenine links with thymine, whereas guanidine links with cytosine. So in a double strand, you can always see the pairing between adenine, thymine, and guanine and cytosine. So these are the structure of this. And when we look into this, a homologous chromosomes, this portion is considered as a gene locus. Gene locus is nothing but the position of the particular gene in the chromosome. Whereas the allele is a part or a single nucleotide, a single base in the gene. So in this case, if you see, there is allele A1, which is somewhat different from the allele A2. So these two are called independently as alleles. In combination of these two are called genotypes. Okay. So allele, a gene can exist in different forms across organisms. How? Because of the change in allele. When a gene has changes in one particular nucleotide, that will assume different forms. So that's why the polymorphism, gene polymorphism. Same gene from you and me will be different with respect to a particular nucleotide change. So that is called allele. So one variant form of a gene at particular locus is known as allele. So in general, a single nucleotide base of a gene segment is considered as allele. So locus, I explained that the exact fixed position or location of the gene on the chromosome is the locus or location, loci or locus. Then, when does allele of same type, if both A1 is there in the two pairs of chromosome, then it is called homozygous allele. If A1 and A2 is existing, then it is heterozygous allele. So the combination of these two alleles are called genotypes. So we will be finding these things in our genetic testing, whether the genotype is heterozygous or homozygous. If it is homozygous, whether it is a wild type or a mutant type or a variant type. So the heterozygous is always a mutant type because that harbors one normal allele and another abnormal allele in terms of disease phenotype. So the organism's phenotype is determined by its genotype. That's why each individuals are different. So when coming into the central dogma, how the DNA gives the message to the RNA, how the RNA gives a message for the making of protein. So this is called central dogma. The DNA is transcribed to mRNA, or the three different RNA called rRNA, tRNA, and mRNA. The mRNA is the actual coding part of the central dogma. So that mRNA gives rise to message for the making of protein. So the proteins can be again classified into two major types: the structural proteins that are responsible for our morphology, anatomy, or bodybuilding, all those things, and the functional, the proteins that are responsible for hormones or enzymes. So here in the table, I have shown the genetic codes and codons. So these are the triplet codes. The triplet codes are responsible for single amino acid. So it adjacent to the code like UU is uridine. So in corresponding to DNA, it is T, thymidine, whereas it will be U in RNA. So TTT or UUU is coding for amino acid called phenylalanine. So like UUA, UUG, like that. So you can have 21 different amino acids. So the amino acids are colored in a way. The non-polar aliphatic amino acids are in red, whereas the polar uncharged. Polar means that will have a kind of directional philicity, hydrophobicity or hydrophilicity. Okay, then positively charged. It can be a positively charged amino acids or negatively charged amino acids. When the amino acid, when a protein has a change with this type of amino acid, say for example, instead of positively charged amino acid, when there is a replacement of negatively charged amino acid due to a mutation, so the functionality of the protein will be affected. Either it may gain of function or loss of function. So the protein may be abundantly functioning or the protein may be silenced. It may not have any function at all. So these are the certain terms. Okay. So when coming to the organization of the gene, so the gene comprises of different portions. It starts with promoter region. This is the region that is responsible for the transcription of the gene or transcription of RNA. So the gene has different elements like coding and non-coding elements. The coding elements are exons, which is in the orange one. The non-coding elements are introns. So essentially, the coding elements or exons are combined with each other to form a functional RNA. Then that functional RNA gives rise to the protein. So anything happens in this exonic region that is going to affect the RNA structure, which in turn subsequently affect the protein. So the expression of these exons are controlled by other elements called untranslated regions. So it is there in both the sides or both the flanking region of the gene called 5' untranslated region or 3' untranslated region. Okay. So then finally there are poly-A signal. So that is the region where the RNA will be terminated. The transcription of RNA will be terminated, and that is the signal for the stopping of the protein synthesis. So the protein will be stopped there. So that is called termination, followed by poly-A nucleotides, poly-A signal. So this is understood. So normally all genes that are transcribed from the 5' to 3' direction. The 5' is nothing but the phosphate group of the DNA strand. So always the nucleotide will have phosphate group at the 5'. 5' means at the 5th carbon. Whereas hydroxyl 3' hydroxyl group. So that is in the so the transcription or translation, the transcription takes place from the 5' to 3' direction. And in promoter region,
Dr. Rajkumar, can I ask you something?
Yes. Yeah.
When we are thinking of genetic change or genetic mutation, where we should be looking at into this drawing if we want to proceed to, for example, the retinal genetics mutation or whatever related?
Yeah. So the mutations can happen anywhere in this gene, the entire area. Like in promoter, there are a lot of promoter variations. If there is any variation in the promoter, the regulation of gene will be hampered. Say for example, if any mutation in this the TATA box, we call it as a TATA box. These are the regulatory element in the promoter. So the proteins that are necessary for the transcription, we call it as the transcription factors. The transcription factors binds at this TATA binds at this TATA box, CAT box or GC rich region. So from there, this regulates the expression of the gene. When there is a mutation in these particular boxes, what will happen? The transcription factors cannot bind. It means the gene will not be expressed. So the gene will be silenced. So say for example, if it is a retinaldehyde gene, which is responsible for retinal metabolism. So the retinal metabolism may not take place. So obviously the retinal function will be affected. So the mutation in promoter is also have major role. And moreover, all these exonic region are also more important. The variation at any of the exons that also will affect the functionality or coding of the protein. The codes will be changed, so thereby the amino acids will be changed. Okay. In some cases, the intronic variations are also important because we call it as a splice site. Normally what happens after the transcription, the exon 1, exon 2, exon 3, exon 4, exon 5 will join together by leaving the introns, or the introns will be excised out. Suppose if there is a mutation in this is called splice site where the introns will be excised out. Suppose when there is a mutation in this splice site, what will happen? Sometimes the splicing will not take place. So we will likely to have a exon with intron 1, then followed by exon 2. So the gene length will be increased, the mRNA length will be increased. You will have non-essential amino acids in between. So this may lead to aberrant protein or abnormal protein. Sometimes the protein will be terminated if there is any stop codon within this intronic region. So the protein will be having only exon 1. The amino acids responsible for exon 1. It will be terminated here. So mutation in all these regions, including promoter, exon, introns, are important nowadays. And the 3' untranslated region and 5' untranslated region are also important because there we have microRNAs. The microRNAs are the regulatory RNAs that all sometimes binds to that and that regulates the expression of the gene. So when there is any change in the translated region, so the microRNA cannot bind or it can bind permanently. So thereby it alters the expression of the gene. So in a nutshell, mutation in any part of the gene may lead to some functional defect. It may be gain of function or loss of function, depending upon the type of variation and the position of the variation. Is it clear? So now, how these mutations have been classified. So what are the different types of mutations? So in a paper by Hussain, so they have classified the mutations into 5 major parts. I mean, not 5, 3 major parts. So point mutation, chromosomal mutation, and copy number variation. The point mutation is nothing but when there is only one single nucleotide change in any part of the gene segment, any single nucleotide variation that is called point mutation. That single nucleotide change may be either substitution. That can substitute one nucleotide by another one. Say for example, if there is adenine, instead of adenine, there could be substitution of guanidine or cytosine or thymidine. So it means this is substitution. Then another point mutation is called insertion. So insertion of any single nucleotide in any portion of the gene segment. So it could be A, T, G, or C. Then deletion. So removal of one nucleotide from the gene segment. Single nucleotide is deletion. So sometimes the insertion deletion can be large. So we call it as a large indels. Insertion, deletion, mutation called large indels. So a segment of nucleotides will be deleted, or segment of nucleotides will be inserted, or segment of nucleotides will be duplicated. The same segment, the sequence will be same, the alphabetical or whatever the order will be the same, that will be duplicated in the same region. Or translocation. That particular duplicated region segment will be located somewhere in the chromosome. So that is called translocation. So these are all called chromosomal mutation because that involves large portion of the gene segment, or copy number variation. Say for example, some particular gene will be in multiple numbers. Say for example, gene A, Huntington's disease, we can call it as a repeats. So same nucleotide set will be repeating. In some cases, you will have same gene will be in multiple times. So that is called copy number variations. That expanding trinucleotide repeat as in Huntington's disease, where you can see CAG repeats, continuous repeats. The repeats dictates the severity of the disease. Okay, so these are the major classification of mutation: point mutation, chromosomal mutation, or copy number variation. So when you take point mutation, we can still sub-classify it into 5 different categories. One is synonymous or silent substitution. Synonymous means, I go back to the previous slide. So here, say for example, you see there are 2 codon for phenylalanine. If this codon, the last nucleotide is changed to C, suppose instead of triple U, when you have last nucleotide as C, so this is not going to change. This is going to give me again phenylalanine. So this kind of mutation is called synonymous variation, or meaning same meaning, synonymous variation or silent variation. It doesn't give any abnormal protein. Whereas if this U is substituted by A, so what will happen? Instead of phenylalanine, I will be having leucine. So this is called missense variation because the reading is missensed. Instead of phenylalanine, there is a replacement of leucine. So this is called, this is called missense variation. This is going to affect the function because the phenylalanine is an aromatic amino acid. The aromatic amino acid is substituted by a non-polar aliphatic amino acid. So definitely there will be a protein dynamics. So the structural changes will affect the function. Then nonsense substitution. So nonsense means any substitution when it is terminating or changing the codon into a termination codon. We have termination codon called, here it is, UGA is a stop codon. UAA, UAG. So these are all called stop codon where there is no amino acid. So at that position, the protein translation will be stopped. Suppose in case of tyrosine, if this U is replaced by A, what will happen? Instead of tyrosine, there will be a stop. So the protein will not synthesis beyond that. So this is called premature termination. So after certain amino acid addition, when the translation machinery come across this particular codon UAA, this will stop. The protein will no more synthesize. So that's why it is called nonsense. So because it doesn't give a meaningful protein. So that is called nonsense protein. Okay. Then
you call that nonsense protein?
What?
You call
Nonsense.
that protein the
Nonsense protein. Nonsense protein because it will not be functional.
Okay.
Yeah. Then the premature termination, which I told more or less same. Nonsense or premature termination. The premature termination could be due to either substitution or insertion. Sometimes what will happen? Again, I'm going back. So instead of this UUU, so I have here UUU. If I have UAA here, again this will change it to stop. So this is substitution. Okay. Then instead of here, tyrosine, if I insert something, A here, if there is insertion of A between A and U, so what will happen? This will change the frame. So this UAA will be considered as a stop codon. So this is insertion. Then in another example, if there is subsequent A, if that is deleted, so that may turn into a termination codon. So the insertion, substitution, or deletion can change a coding codon to a non-codon or stop codon. So these are all called frame shift. So frame shift mutations. So that will shifting the frame, coding frame. So after some region, there will be a normal protein sequence. When there is an insertion or deletion, the frame shifts. So there will be alteration of amino acid. So from there onwards, the entire fragment will be changing. So this is called frame shift mutation. It may be a complete coding protein or functional protein, or it could be a premature protein, depending upon the type of deletion or insertion and the location of the insertion or deletion. So, how to classify the mutation? Because not all mutations are pathogenic. Okay, we may harbor different mutations in our genome. Can we consider all mutations as a pathogenic? No. Some mutations are beneficial. Some mutations are going to cause a disease. Some mutations are going to help us to survive this environment. Some mutations are going to benefit out of mosquito bite. So these are the changes that we have because of the mutation. So the mutations can be classified either benign, like benign means there is no change, or there is no significant functional changes with the protein. Likely pathogenic. So it could cause pathogenicity. Or pathogenic, definitely it is going to cause the disease or abnormality. Or uncertain significance. It means those genes or that particular mutation is not validated by the experimental procedure. So all these likely pathogenic have been experimentally proved by animal models or a large pedigree with a different family for the particular variation, particular mutation. If that is causing the same disease, then this is established variation. So that can be classified as either likely pathogenic or pathogenic. If any variation or gene is not evaluated or validated with a different population or with a different family or in any of the experimental model,
It got frozen for a while. Can you hear?
yeah, it is unmute, yeah. Can you hear me now? Yeah. Okay. So where I left, have you followed all?
Yes, sir.
Yes, it's all right now. Yeah.
Okay, fine. Yeah. Okay. So the sequence variations can be classified into 4 types: benign, likely pathogenic, or uncertain sequence. Okay. Now coming to the family history. So how much the family history is important in determining the inheritance pattern as well as identifying the gene of interest. Okay, so we need to collect the entire family history from the patient. So their grandparents, grandparents, all those history needs to be documented to establish the pattern of inheritance. Then in addition, So this family history will help us in eliminating certain cases where the environment plays major role. Say for example, polygenic diseases are multigenic or multifactorial diseases can be excluded from the genetic study if you know the entire family history. So most of the genes that may harbor either mutation or polymorphisms, okay, that could be the reason that is inheriting or segregating in the population. If it is a single gene mutation, so that can be easily captured when you have a strong family history. So based on that expression of that mutated allele, so we can call it as either dominant or codominant or recessive. So depending upon the number of pairs. So the family history will tell us a kind of clue that what we can expect when we go for the genetic testing or how we can target that particular gene of interest. Okay, so probably you might come across all these symbols, but still how to make a pedigree will be a bit complicated task because drawing a big family pedigree will be a bit cumbersome. One generation, two generation, or single family, two family is a bit easy, but multiples of families with different more than three generation is a bit difficult while drawing manually. So there are softwares available where we can select the symbols and we can just annotate it. So that will make a pedigree of our choice. Okay. So in general, so the male is symbolized by squares, open square, whereas female by open circle, and the marriage between male and female is indicated by the link. When there are two links, it means it is consanguineous marriage. That indicates consanguineous marriage. When the link is broken, it means it indicates divorce. Okay. In any individual, if it is crossed, so it means deceased. Both male and female are applicable. It is deceased, and when the box or circle is filled, it indicates the phenotype or disease phenotype. They are affected, the affected condition. The arrow adjacent to that indicates the index case or the probands. Okay. Then the twins are indicated by split split arrows, whereas the identical twins are shown by connecting between the twins. When there is ongoing pregnancy, if the gender is not known, then you can have a diamond shape. When there is a carrier, there will be a dot inside. When the gender is not unknown, not known, then it is a diagonal or a diamond. When there is an adapted kid, the symbol will be like this. When there is no offspring, you can have one large and a small lines. And when there is a stillbirth, only one single dot. So these are the certain symbols that is commonly used in the pedigree making. Okay. So let us look into different patterns of inheritance. So take the first autosomal dominant inheritance, where you can see almost all, not all generation has affected members. Whereas in autosomal recessive, in the first generation you can see, whereas in the second generation there is no affected member, in the third generation it reappears. So this indicates autosomal recessive. And in X-linked dominant, if you see the female is affected here, so obviously all gender in the subsequent generation will be affected. And in case of X-linked recessive, so the female that pass on that affected chromosome to males only. So this passes from female to male. Whereas in mitochondrial inheritance, you can see the female that pass on that defective mitochondria to all the members of the family. Okay. So now how much the gene is been shared between the relatives? So we can classify the relatives into three groups: a first generation relative, or second generation relative, and third generation relative. So the first generation relative comprises of mother, father, full sibling, and the child. So this will be the first generation. Then the second generation, you'll have your grandparents, grandchildren, half sibling, aunt, uncle, niece, nephew, all are second generation. Whereas the third degree relative are your first cousins, great grandchildren, or great grandparents, or great aunts. So in the first within the first degree of relative, you share about 50% of your genes. So the gene will be shared in 50 percentage, whereas in the with the second degree relative, there will be 25% of genes sharing. Whereas in the third degree relative, you will share 12.5% of genes. So you can have a, I'll show you the conjunct relationship chart where you can easily find
Yeah, the sound.
yeah, now, yeah, it
for a
is
while.
good.
Okay.
Am I audible now? Yeah. Okay. So myself, I'm sharing 50% of my gene with my parent and 25 with my grandparents, 12.5 with my great grandparents, and the second great grandparents of about 6.25 and 3.125. So the gene sharing will be reducing by half as and when the generation increases. So similar way, when you pass on to that, it will be decreasing. So this is a general a kind of ready reckoner to understand the degree of relatives and the amount of genes that is shared between different relatives. Okay. Now the question is, we have a patient, so we know that he is having a particular condition. Now how to do that? So by clinical evaluation, we identify the phenotype. Now we know the phenotype. So what could be the reason for that phenotype? How to identify that phenotype, the causation of the phenotype. So for that, we have different genetic testing methods. The simple one is called PCR-based Sanger sequencing. So this was the gold standard method technique which people were using for the past 30 years or 3 decades. Now with the advent of next generation sequencing, so we can go for high throughput analysis. Whereas with the PCR and Sanger sequencing, you can go maximum of 8 or 16 samples with that machine. Now with the next generation sequencing, we can go more than 96 samples together. Now certain platforms are available, even you can go for 300, 384 samples together. So, and moreover, the sequencing of our chromosome, our genome, is very robust nowadays. Whereas with the Sanger sequencing, it is time consuming. But now it is very fast. Within a period of 2-3 days, you can have your entire whole genome sequencing. So that is the advantage of our next generation sequencing. So there are different methods available based on the cost effectiveness as well and the applicability. So certain equipments we can host in our institute, certain we have to outsource it because we cannot afford buying that kind of equipment like a next generation sequencing. Okay. So this is a study from my previous hospital rather. So we received a case. Actually, the case has congenital cirrhosis along with glaucoma, cataract, aniridia, myopia. So it is a multifactorial kind of multifactorial diseases. And when you look into the pedigree, the mother had the same phenotype, the brother had the same phenotype, and the proband also had the same phenotype. whereas the father did not have, but he was blind since birth. So we collected blood sample from all the patients and we did Sanger sequencing. And the adjacent C, the C indicates the chromato, we call it as a chromatogram, the sequence chromatogram. This will show the change in the nucleotide. Here you can see the C, father, the T, the red one is T, blue one is C, the red one again T, and the green is A, and the black one is G. So you can read the nucleotide sequence T, C, T, AAT, CGA, and AG. The TCT is responsible for amino acid called serine, and AAT is responsible for amino acid called asparagine, and the CGA is responsible for arginine. And in the proband, if you see proband and brother, there is serine followed by arginine, whereas here it is TGA. As I told, the U is T here. So if you consider UGA is stop codon. So it leads to termination. So instead of R, there is a substitution of termination codon. So this protein will be terminated. So we call it as R261X. The arginine at amino acid position 261 is terminated. So it means the protein is incomplete. So where it is incomplete, this is the entire structure of the protein. The protein has different domains, NTS, CTS, HD, and PSDD domain. The change in amino acid here that ends the protein sequence by here. So you will have only this portion of the protein. This portion will not express in the actual protein. So the protein will be non-functional. So this is what we observed in a case with multiple phenotypes. And what we hypothesized here is this conjunctival cirrhosis is a disease associated with vitamin A or retinoic acid deficiency or insufficiency. And we hypothesized that since this PAC6 gene, this gene what we took is a PAC6. This PAC6 is responsible for regulating a metabolic gene called CYP26B1, which is responsible for metabolism of retinoic acid. So what we hypothesize, when there is a defect with PAC6, the PAC6 cannot regulate the expression of CYP26B1. In the absence of CYP26B1, the retinoic acid will not form. So the retinoic acid, in the absence of retinoic acid, you will have cirrhosis because vitamin A metabolism will not be there. So there will be a congenital cirrhosis. So this particular patient had a supplement of vitamin A, 2 lakh unit, so 2000 units in single dose, and he was advised to have it for until he reaches 5 years of age. So now the patient is okay. So he is cleared of this congenital cirrhosis condition. So this is the application that what we had from the genetic testing and subsequent therapeutics. Of course, we do have a lot of limitations in genetic diagnostics. So the major two reasons are genetic heterogeneity and phenotypic heterogeneity. When there is genetic heterogeneity, it means the parents may have a certain type of variation, and the children may have another type of set of variations. So when there is a genetic heterogeneity, obviously the degree of that disease within that family will be different. So the phenotypic changes will be there and the degree of severity will be different. So all those things will always happen as in our previous case. So wherein here the mother did not have a particular condition called, I think the mother did not have cataract. Only these two children had subluxated lenses. The mother did not have that. So these are the things. So that could be because of the genetic heterogeneity or the phenotypic heterogeneity. Because sometimes the clinicians may not express the phenotype or write the phenotype uniformly. The diagnosis will be different with different clinicians. The way how one clinician perceives that particular phenotype will be different from the other clinician. So there will be a phenotypic heterogeneity. When that occurs, alternatively, that will affect the genetic heterogeneity and the genetic testing. We may not be able to give a pinpointed genetic diagnosis when there is a phenotypic heterogeneity. Am I clear?
Very clear, very clear. And you gave such a very great lecture, and you actually said everything, you know. It's, I didn't know it that was so complicated, even though we had this idea, but it's difficult to do genetic counseling, for example, because you have to involve all this information and gather everything and to get to the diagnosis and to compare to the clinical backgrounds. So
Yes.
not
Yeah.
like this, yeah. And you're
Yeah.
explaining very well.
Yes. So you need to have a strong genetic counselor who can who has the capacity to draw all the information from the family. Other than the clinical evaluation, we need to have additional information from the family. Then it will help the genetic diagnosis or the genetic diagnostic people in a better way. So they can pinpoint that exact causative variation. Otherwise, it will be like a gross observation only. So these are the genes, or this gene may play a role, or this particular variation may play a role. All those probabilities only they can say. They cannot pinpoint the exact causative variation.
And how many people are included in your genetic team? I'm not including the residents and the fellows, but I mean your team
So
for.
currently we are three. We are three members in the genetic unit. So I have two, my colleague, they are kind of genetic counseling as well as technician, laboratory technicians. So one is already working, I mean, she is doing her PhD in neurodegenerative diseases and eye diseases, something related to that.
Yeah, very good. We learned a lot from everything, and especially from our previous chat as well. And I already told our residents and fellows that we are gonna include your lecture as well in our resident test,
Okay.
next text.
Okay. Oh, thank you.
So they're gonna read it up and go over your slides again and your referrals, references. That was
Yes.
great.
Yeah.
I surely appreciate, and I don't think you could get anything better because that was perfect. You know, you covered
Thank you, sir.
all issues on genetics. And of course, if we have to extend on that, we could probably take 4 or 5 hours and talking and discussing
Yes.
different cases. And
Yes. Yes.
we are going to do
Yeah.
that
Yeah, we
later
will do it
on.
one by one. Yeah, we'll do one by one. We will take one particular case or one particular phenotype so we can talk about it. So that's what I wanted to keep very simple here because I don't want to bring in so many things which will make confusion at the end. So I wanted
Yes.
to keep it very simple. So we'll go one by one like next presentation we can have a kind of RP and the genetics, and in another session we can have AMD and genetics, something like that. So people will also understand what are the genes responsible for RP, what are the variations are responsible for RP, all those things.
All right. And
Yeah.
this lecture is like a landmark for the things, you know, because from these, people would get an idea of what to look for and also to what to study and then to ask. And so we could direct later on in the next programmed visits some cases as well, and we're going to join some cases of ours that we could discuss. And I really appreciate Rajkumar again for your participation.
Thank you so much.
Thank you so much. And we are open for any discussions from anybody. Aishwarya, do you want to comment on anything?
Thank you. First of all, thank you, sir. That was a very nice talk. I just have one doubt, sir.
Yeah.
So when a patient comes to the hospital and we suspect a genetic disease, how can we tell, for us lay people, I mean, in terms of genetics, we are ophthalmologists, but we don't have the vast genetic knowledge that you might have. So how can we recommend what type of genetic testing we should advise this patient? Is there a format or some sort of template that we can follow?
Yeah, Siddhi has all the information. Okay, so for your information, if you want, we have certain templates from Genome. That is a company where we do genetic testing. Okay, it depends
that applies
upon
to all the genetics diseases.
Yeah, it depends upon it depends upon. Normally, they go for only one particular panel called retinal degenerative panel. So that comprises of around 700 genes. So they will sequence all the genes and they will find out the gene that is responsible for that particular phenotype. So you can have the data like this. I think I have shown that. Yeah, here. So in this picture, you can see. So here, these are from different phenotypes like retinitis pigmentosa type 3, chondrodystrophy, Stargardt disease, retinal pigmentosa 41, retinal macular dystrophy or 41, Joubert syndrome, Stargardt retinitis pigmentosa 31. So these are all the different phenotypes we got, and we submitted all the samples to the service provider, and they did sequencing for almost 700 genes. I forgot to keep that gene list. In next session, probably I will share the genes. So they found in these particular genes. And if you look into the gene set, the PROM ABCA4 genes are repeated in so many cases. So they will give a report like this. You can see hemizygous. So hemizygous indicates it is a sex chromosome, right? So it is X-linked.
Yes, sir.
Okay. So the homozygous, so mutation is in both the chromosome. Heterozygous, one chromosome is okay, one chromosome is affected. But it is a composite. The same gene in two different positions, say for example, exon 42 and exon 48 has two different mutations. Though this is called composite heterozygous mutation. That's why it has undergone under autosomal recessive category.
Okay.
But still, it is likely pathogenic. It is not pathogenic. It is likely pathogenic. The effect of that variation is not understood much. Okay, so this kind of report.
Yeah. Sorry, sir, is homozygosity and heterozygosity linked to the progression and visual prognosis in any way?
Yes, yes. When there is a homozygous defect, what happens? Both the chromosomes are affected. Right. Suppose if it is a loss of function due to that mutation, so it means both the chromosome cannot give a functional protein. If the protein is very functional, it is essential for the maintenance or sustenance of the tissue, then the tissue may not maintain its liveliness. You got my point? So yes, homozygous has more severity than the heterozygous. In heterozygous, what happens? One chromosome is abnormal, one chromosome is normal. So the functional defect is compensated by the normal chromosome. So there will be some activity of the protein. Some 50% activity can be expected from the normal chromosome. So the tissue can sustain the condition for some time. So the severity will be automatically less when compared to homozygous condition.
Okay.
You got it?
Yes, sir.
Yeah. Okay.
Hi, Jaydeep. Hi, Jaydeep. I think you.
Hello. Hi. Hi. Good morning, Dr. Hudson.
Good evening.
Good evening, Dr. Rajkumar. Yeah, what a fantastic lecture. I'm sorry I couldn't join at the beginning. I was at the another branch clinic, and absolutely no range there for internet.
Oh, I see.
So
Oh,
even
okay.
right now I'm driving. I'm driving and I'm listening to your lecture.
Oh, I see. Oh, thank you. Thanks a lot.
So it was really comprehensive, and I couldn't hear the first part, but it was really good. And I think we need to keep having this kind of a refresher course in genetics every 6 months, because we tend to keep on forgetting
Yes, yes,
genetics.
yeah.
So it's something
Yes,
which
yeah.
is very easily volatile for the
Yes,
memory.
yeah.
But
Yeah.
fantastic, you put it very well, and I think you should share your presentation with us so that we can, keep referring to it. At times we don't, remember
Yeah,
things.
yeah, sure.
So
Yes, sure.
it
Yeah,
looks
yeah.
like a very good ready reckoner for us to refer.
Yeah. Thank you. I think it is in the
And
YouTube now, right? It is shared in the
Yes, sir.
YouTube. Yeah.
Yeah, yes, we're gonna
right,
leave it in YouTube. I'm gonna get the Zoom version that is
Okay.
much better. And
Okay.
anybody else would like to ask, and like to add anything?
yeah, I think we can,
And
as
I wanna thank.
hello.
Hello.
Yeah, yeah. Sorry, I said that. Yeah, I think it's a good idea to keep one lecture on genetics every month or so, and we can
Yes,
take
yeah. Yeah.
take it slowly, one step at a time, as you said.
Yes. Yes. Yeah. Yeah, we can go
That's true.
slowly because the understanding concept needs lot of time. So we
Yes,
will
Yeah,
sir.
yeah,
go very slowly so that everyone should
Otherwise,
understand
people
and they
get
should.
confused. Otherwise, people get
Yeah.
yeah.
confused. And
Yeah, probably
yeah,
we need to come prepared. Probably we need to read up and come.
Yes.
all right. So I'm gonna close the
So,
session. And,
Okay.
I
Yeah.
thank
Yeah.
everybody again for participating. This is
sure.
gonna be on YouTube, and it's gonna be there for a while in a live streaming. But then I will download the Zoom version that you have the better images, and later today it's gonna be there. Thank you very much
Okay.
again, and
Thank you.
have a great day for
Thank
all.
you. Thank you, everyone.
Yeah. Thank
Thank
you
you
all.
so much,
Thank you
sir.
all. Yeah.
Bye.
Thank you.
Bye. Bye.
Yeah. Thank you. Thank you. Bye bye.
Bye, bye,
Bye bye.
Bye.
bye.